Koch's lab, focusing on fisheries ecology. So there's some different challenges with wildlife ecology and how you go about these studies, particularly where do you find these animals and how confident are you in your sampling that you haven't missed them. So this sign is very useful. I thought it was very useful. It lets you know that that room is occupied and that there's some very important business happening there. We don't have these types of signs on the landscape. Sorry, go back. To let us know which streams are occupied in a watershed and which ones have important habitat for those species. Fish move, we know this, and so we have to, they use different parts of the streams at different times, so we need to, once we pick a stream and we start sampling, know that we didn't miss them, have confidence in our process there. So I'm not going to be focusing on salmon, but the salmon family, the salmonid family, Arctic grayling in the Little Nahanni River watershed, which is in the southwest corner of the Northwest Territories. The Little Nahanni River is up there. It overlaps, its watershed overlaps with the Nahanni National Park as well as the Nazi Cho National Park, which is a new park in 2008 that was developed up in the upper part of the Nahanni watershed. So why do we care? Why are we studying Arctic grayling? The resource managers in the north have identified, stream salmonids, the assessment and monitoring of stream salmonids as a priority to understand their vulnerability to climate change. There's increasing development pressures in the north. So we need to know where they are, how they're doing, what are the trends in order to assess what the impacts may be. But there's a lack of information out there in the north, as was previously said, on where we find species. So there's distributional monitoring is an approach that's gaining ground. It's you look at the distribution of a species over a landscape in suitable habitat patches. So here's an example of where you can, within a stream and within a watershed, try to identify where these suitable habitat patches are. There's been work on this on juvenile bull trout in the northwest US, but there's an interest in looking at it as an efficient and rigorous approach that can be used in vast and remote areas to go in and find out what's there and have confidence that you didn't miss them. Type 2 errors, essentially, we're trying to avoid with some of the sampling. So can this approach be used? Do we understand for Arctic grayling specifically, because they are found in a lot of these watersheds? Do we understand enough about their habitat? Are we looking for the right signs on the landscape to identify their suitable habitat? And then to go out there, the best way to delineate suitable habitat is do direct observations. So we're going out there and doing that. So what is a distributional monitoring approach? You take your study area, the Little Nahanni River watershed, you define what your criteria you think might be at that watershed scale to try to delineate suitable habitat. These are the criteria that were used. Select your streams. So these are the top 40 streams that were selected within the Little Nahanni watershed. Then you use a random process to select your sites within the stream. It was called generalized random tessellation stratified design or something that just essentially stratifies. I'm really off on this one. You can tell I didn't practice enough. Spatially representative within each stream length, and each site is 100 meters. So you hit the ground with your GPS and your maps, and each site is 100 meter length. We are sampling for fish presence. Are the grayling there? That's our main response variable that we're looking for. You do electrofishing surveys, then you collect various habitat variables that are some of your predictor variables in that site, and then you go out and repeat your fish surveys within the same sampling period as a way to make sure you weren't false absences or type 2 errors. So here's some of the data we collected on water velocity and slope and what it looks like. Some of the stuff I put up, not pretty. I did some histograms, ran some means and standard deviations. It was kind of messy in a lot of instances, but I didn't do anything to fix it up yet. Just made some pretty diagrams. So what we ended up being able to actually achieve out there was we sampled 109 sites and only found grayling in 8. So that's not great for detection. It's not great for stats either. We have an N of 101 for sites without grayling and an N of 8 for sites with grayling. Another thing I just want to point out is patch 10 was a stream where we did find grayling. First time we went out and sampled, nothing there. Second time we went out and sampled, we found them. So sort of reinforcing that, why you need to go out again. Here's some of the data once we did catch grayling. We took length. So this gives you just a length frequency distribution is a way we display this. Most of them were found in 2 patches, 128 and 67, and we caught a lot of small ones. electrofishing targets smaller fish, but it's kind of just points out that we may be missing some habitat information on the larger fish if we're only catching the small. I went there, I went crazy with the scatter plots to look at the various stream channel characteristics, because they all kind of relate, instead of looking at one and this and that, to kind of as ways to look at relationships between some of these stream channel characteristics and what that might mean for grayling. So the red dots are grayling. Some you did see some correlation, depth and velocity, slope and velocity. Where you think you'd have a bit more correlation, there wasn't, but things like slope, you started to see some trends where you may think that that's where the grayling sites are at the lower end, so we're starting to see some patterns, potentially. Here's some of the habitat characteristics that we did. Each one, substrate and habitat types were characterized into 4 groups. It was dominant, so we looked at percentage, so percentages on the y-axis of these various characteristics. For the sites where grayling were present and absent, so 0, 1, presence, absence. You can see it starts to give you a little bit of information. It's messy, a little bit of information on they prefer silty sand and gravel in the sites that we had, riffles or pools and runs over riffles and cascades. So just trying to establish some of those characteristics. And then I looked at some of the major biophysical characteristics that we're looking at the landscape. So elevation, slope, water temperature. Some of those things we can look at a broader scale to identify where they are. So there's elevation and temperature in the far one, and stream width and channel slope. Black dots are where we found the grayling, white dots is where we didn't. And then the red line is the mean for all the sites. The green lines are the means for the grayling sites. So I just was using this as a way to try to see, well look, in this one, in channel slope, the means for grayling was quite a bit smaller than what we were looking at our broader criteria and all the sites. Elevation was lower, as well as water temperature higher. So it's starting to pull out some trends. So we did a whole lot of walking and not a lot of shocking, as one of the people in the crew did. We had 2 crews, went out 3 times over the summer. I only presented on the first one, but I'm hoping by getting the rest of the data together, I'll get some more information on the grayling sites to look at. And right away, I mean, we saw the benefit of having more detailed criteria, because we were hitting all these sites, not getting grayling. This looks great. We had fun going up in these streams, but you're at 1,500 meters, 1 degree water with only 10 centimeters. You're not going to be getting grayling. So it's not efficient at this point as a process to try to go forward with, but lots more to learn about grayling ecology. Help us sort of narrow that down. There's going to be some more sampling happening in the summer. So I know I went way over. Sorry. Thanks.